How do you keep a really delicate chemical system perfectly balanced? Whether we're talking about the blood in our own veins or, a multi-million-dollar experiment in a lab, so much of life and science really depends on stability. So what happens when that balance gets thrown off?
Well, today we're going to dive deep into the unsung hero that makes all of this stability possible, the buffer solution. And honestly, it might just be the most important chemical you've never really thought about.
The whole story of buffers really starts with this quest for stability. And the main villain in that story, it's pH. You've heard of it, right? It's that measure of acidity or alkalinity, and it is a super critical variable in pretty much every chemical reaction out there.
And when I say critical, I absolutely mean it. Even a tiny, tiny little shift in pH can be a total disaster. It can completely tank a sensitive experiment, it can make a drug totally useless, or even worse, toxic. So yeah, the stakes are incredibly high.
So how in the world do scientists keep this really volatile variable in check? Well, they bring in the hero of our story, the buffer solution. Think of it like a chemical shock absorber. To put it simply, a buffer is a solution that just says no to pH change. You can add a little bit of acid, you can add a little bit of base, and that buffer will fight like heck to hold the pH steady right at the level you want it to be.
So how does it pull this off? Well, it's all about equilibrium. See, a buffer has two parts, a weak acid and its partner, which we call a conjugate base. Think of them like a tag team. If some outside acid comes along and tries to mess things up, the base partner jumps in and neutralizes it. If a base shows up instead, the acid partner steps in to handle it. It's this perfectly balanced, self-regulating system. It's amazing.
Okay, so that's the theory. Now let's see how this stuff actually works in the real world. Because in the lab, buffers go from being this neat concept to an absolutely essential tool for precision and purity.
And you want to know their most common, most fundamental job? Calibrating the very instruments we use to measure pH in the first place. I mean, think about it. If your pH meter isn't accurate, then none of your other measurements will be either. Buffers are literally the bedrock of reliable data. And scientists are, super rigorous about this. For maximum precision, they don't just check against 1 or 2 points, no. They use what's called a 5-point calibration. By measuring the voltage across 5 different super accurate buffer solutions, they can map out this precise calibration curve. It ensures accuracy across a whole range of pH values. This is the gold standard for sure.
But how do you know your buffer is accurate? Well, the answer is something called traceability. A certificate like this one shows that the buffer's value is directly traceable to a master standard from an authority like NIST. That's the National Institute of Standards and Technology. It creates this chain of trust that underpins all scientific measurement.
Okay, so even with a perfect buffer, there's another problem. Us, humans. Every single time you open a big bottle of buffer, you risk contaminating it. The solution? These single-use doses. It's brilliant, right? This guarantees that every single calibration uses a perfectly fresh, totally uncontaminated, and accurate standard. All right, so we've established that buffers are kind of like the pH police, right? They keep everything stable. But what if I told you that for some of the most important molecules in medicine, I'm talking about proteins, that is only just the beginning of the story?
This quote from a really important research paper says it all. For proteins, choosing a buffer is a formidable challenge. So why? Why is it so hard? Because the buffer stops being a simple background character and starts playing a leading role.
This is where the whole game changes. With proteins, the buffer doesn't just sit on the sidelines maintaining pH. No way. It actually interacts with the protein molecule itself. And the wrong buffer? It can cause a protein to unfold, to clump together, or to lose its function completely. It can literally destroy it. So let's pull back the curtain on a buffer's secret life. This is where things get really interesting, and we see just how surprisingly complex and influential these solutions can be.
Okay, take freeze drying. It's a really common way to preserve protein-based drugs. When you freeze a sodium phosphate buffer, its different parts can crystallize at different temperatures. This creates these tiny pockets of extreme pH that just shred the protein. But, and this is the cool part, if you just swap the sodium for potassium, you can prevent this from happening and protect the drug. It's a tiny change with a massive, massive impact.
Buffers can also interact with the water around a protein. Some of them, called cosmotropes, are water structure makers. They basically organize the water molecules into this stable shell around the protein, kind of giving it a protective hug. But others, called chaotropes, are water structure breakers. They just create chaos, disrupting that protective shell and leaving the protein exposed and unstable. So, choosing a cosmotropic buffer is often a key strategy for stability.
And that's not all. Some buffers have another hidden talent. They can act as bodyguards. They literally hunt down and neutralize these damaging things called free radicals, protecting the protein from oxidative damage, which is another huge way these molecules can break down.
And if you need proof of just how much this all matters, just look at this number. Scientists took an enzyme and found its half-life. That's the time it takes for half of it to degrade. Was only 44 minutes in a standard phosphate buffer. But just by switching to a different buffer, glycine NaOH, its half-life shot up to 938 minutes. That is a more than 20-fold increase in stability, just from changing the buffer. Wow.
So where does this whole journey leave us?
Well, it elevates the humble buffer from just a simple lab commodity to a true unsung hero of science.
I mean, we've seen it go from being a simple shock absorber for pH to this complex, active participant in the delicate dance of biochemistry, stabilizing water, fighting off free radicals, and directly shaping the fate of our most advanced medicines.
And it kind of leaves us with one final thought. We celebrate the big discoveries and the brilliant scientists, but how many of those breakthroughs, from basic lab research all the way to the drug that could save a life, were only possible because someone, somewhere, took on that formidable challenge and found the perfect, silent, stabilizing buffer? Makes you think, doesn't it?